Okay, so hello guys. Now we're going to talk about our development of the nervous system. So the development of the nervous system begins in the third week, and this is from a thickened area of the ectoderm. So this is the thickened area of the ectoderm, and underneath this we have our notochordal process. And this notochordal process is going to induce this ectoderm to start differentiating into the neural plate. So again, this is what starts the entire process of the nervous system. We have our notochord process, which is going to be underneath our ectoderm, and this is going to induce the ectoderm to differentiate into our neural plate. Now, what is our neural plate? Our neural plate is going to consist of our neural groove, as you guys see here, and our neural folds. Our neural folds will then fuse into a neural tube, which is this blue structure, and it's going to detach from the ectoderm. After it detaches, you have some neuroectodermal cells that are remaining, and these are going to form our neural crest. So again, we start with the notochordal process, which is inducing this ectoderm to differentiate into our neural plate, which is what it looks like. This is our neural plate. Our neural plate is going to have the groove and the folds. These two parts will then fuse and they're going to close, okay? They're going to close and they're going to form this blue circle, and they're then going to detach from the ectoderm. So once this blue circle, once these two parts fold, once these two parts fuse, this entire ectodermal part is still going to remain on top, which is what you see here. And in between your neuroectoderm and your neural fold, you have some neuroectodermal cells that are going to remain. And these are going to form your neural crest, which we will later talk about what they are. Now, why is this important, or what is this neural tube, guys? Well, the neural tube is basically what's going to turn into your brain and your spinal cord. And you guys are seeing it from like a cut version, right? This is how it would actually look like. So this entire thing is your neural tube. And your neural tube is fusing opposite of your somites, which are, I believe, are your 4 to 6 somites. So again, this is your neural tube, this entire thing. And it's here. So you're going to have your anterior neuropore and your posterior neuropore. Just so you guys understand what you're looking at, this is going to turn into your brain, and this is going to turn into your spinal cord. And this is all coming from this, from your notochordal process, your ectoderm, forming your neural plate, which is going to turn into your neural tube. You're going to have your anterior neuropore. This is going to close on about the 25th day, and your posterior neuropore, which will close about on the 27th day. And as these neuropores close, this is also going to coincide with the establishment of a blood vascular circulation of your neural tube. Now, let's just quickly go into the histogenesis, or what are the cells that are present, what is going on from a histological perspective. So you're going to have your neuroepithelial cells, right? These are going to be your neuroepithelial cells, and they're going to proliferate into simple columnar epithelium of the neural tube. Gradually, as this is happening, they're going to turn from simple columnar into pseudostratified columnar. Now, this is something that might be difficult to see, but it's just important that you guys know that as the neuroepithelial cells proliferate, simple columnar epithelium of the neural tube, they gradually will change to pseudostratified columnar epithelium. Now, you're going to have these basal processes of these neuroepithelial cells, and the basal processes are going to connect, and they're going to form your inner limiting membrane. So here you have your inner limiting membrane, which is formed from the basal process of these neuroepithelial cells. And this is going to provide support and structure for the proliferating cells. The apical process is going to form your outer limiting membrane. And as the number of your neuroepithelial cells increase, as they start to proliferate, they cannot anchor themselves properly. So they start migrating to what we call the intermediate zone, which is going to be composed of apolar neuroblasts. So again, we have your inner limiting membrane, which is due to the basal process of the cells. You have your outer limiting membrane, which is due to the apical process. And this is providing support. And in between, as they start proliferating, they cannot be anchored to the inner limiting membrane anymore. So they're going to migrate into what we call the intermediate zone. Now, what are some of the cell types of the neural tube? Again, neural tube. We're still in the neural tube, which is going to form your brain and your spinal cord. So some of the cells you see, we have our radioglia. These are elongated cells that have slim processes, and they're going to cover the entire thickness of the neural tube. They act as a guide for the neuroblasts to their destination. So think of the radioglia like a ladder. They're basically going to guide the neuroblasts into their final destination. The neuroblasts, these are cells that are generated in the ventricular zone, and they're formed by your neuroepithelial stem cells. So you're going to have your neuroblasts that are going to kind of be climbing and using your radioglia as a ladder to get to where they want. You're going to see two types of neuroblasts. You're going to see bipolar neuroblasts. These are your cells which are going to be migrating, and you're going to see your multipolar neuroblasts. These are what they're going to change into once they've reached their final destination. You're also going to have your glioblasts, and these are going to be your precursors for your glial cells. Now, another important thing that you guys need to know is that there's programmed cell death, and the programmed cell death is very important. You might see these cells with pycnotic nuclei, and these are cells that are undergoing cell death, and they're broken into apoptotic bodies, and then they're going to be phagocytosed. Why is programmed cell death important? Well, during neurogenesis, right, which is when you have the formation of your neural tube. So again, we're still in the neural tube. So during neurogenesis, there are a lot of neuroblasts that are going to be produced. During the course of synaptogenesis, some of these neurons are going to be able to contact the target tissue, and the target tissue is going to give them trophic support. The neurons that were unable to contact their target tissue will not have trophic support and are going to undergo cell death. And this way, there's going to be a normal number of neuroblasts, and this is also required for the closure of the hindbrain of neural tube, which you guys will see later. So once again, neural tube, ectoderm, notochordal process that induced this entire formation, and then you have some cells that remained, which were forming your neural crest. So now let's talk about the neural crest. Well, the neural crest contains these neural crest stem cells, which are multipotent and they're highly plastic. These cells are going to undergo mesenchymal transition. Basically, the basement membrane is going to break down, allowing them to escape, and then these cells will migrate along the nerve fibers to their final destination where they will proliferate. Now, what are the derivatives of the neural crest? What do the neural crests turn into? We know that this is going to make your brain and spinal cord. So what is this going to make? Well, here are the derivatives of the neural crest. We have PNS derivatives, mesenchymal derivatives, and other derivatives. And because the neural crest can make so much, sometimes it's referred to as your fourth germ layer. So PNS derivatives is responsible for neurons in the sensory ganglia of spinal cord, ganglia and plexus, the adrenal medulla, Schwann cells, satellite cells. Mesenchymal derivatives are leptomeninges, pharyngeal arches, some connective tissue components, which you guys know, cornea of eye, tooth papilla, even your cardiac semilunar valves. Mesenchymal derivatives, you guys know very well mesenchymal derivatives. And then some other derivatives, such as your parafollicular cells of your thyroid gland, your neurocirculatory cells, and more. I was only able to include some of them, but definitely know that the derivatives of the neural crest, especially pertaining your mesenchymal derivatives, it's a lot. Now we're going to talk about our spinal cord. So remember, the spinal cord comes from the top. The bottom, the caudal part of your neural tube. The top is where you're going to have your brain. And so essentially, these are images that are in your lectures, and you guys just need to know what everything represents, okay? This blue represents your white matter. Your gray matter is represented by these, well, they call it, you have your alar plate and your basal plate, and your basal plates, they're later going to fuse and join together. And then you're going to have your ventricle or ventricular zone, and these are going to be your ependymal cells. So later on, around the sixth, so this is all happening around the 5th week. Around the 6th week, you're going to have fusure of your alar plates and your basal plates. Your alar plates are going to represent your dorsal gray horns, and your basal plates are going to represent your ventral gray horns. And you will also see some pseudo unipolar nerves that are derived from your neural crest cells. Now we're going to talk about, so this was the caudal part of your neural tube. Now let's talk about the cranial part of your neural tube. And you need to know these images very well. It's not that complicated. We start with 3 vesicles, okay? So this is the brain, but first it starts off as these 3 vesicles. You have your forebrain, your midbrain, and your hindbrain, also known as prosencephalon, mesencephalon, and rhombencephalon. Around the 5th week, your prosencephalon and your rhombencephalon, or your forebrain and your hindbrain, are going to further divide into 2. So in the end, we end up with 5 brain vesicles. So your prosencephalon is going to turn into ptelin and diencephalon. Your mesencephalon, or your midbrain, is going to remain as 1. And then your rhombencephalon, or your hindbrain, is going to further differentiate into metin and mielencephalon. I know it's a bit difficult. It's easy to confuse some of these words, but yeah, you just are going to have to memorize it. Now, what happens is during the 4th week, as your brain is growing, it's going to bend, okay? So your forebrain is going to bend down, and your hindbrain is going to kind of like bend a little bit up. It's just going to bend, and this is how it's going to look like. So here you have your prosencephalon, or your forebrain. It bends down. You have your mesencephalon, and then you have your hindbrain, or your rhombencephalon. And as it's bending, it's going to create these 2 flexures. This is going to be your midbrain flexure, and this is going to be your cervical flexure. So again, prosencephalon, forebrain, mesencephalon, rhombencephalon, or hindbrain, you have your midbrain flexure and your cervical flexure. As the brain then starts growing, some parts are going to grow more than the others. This is going to cause there's going to be unequal growth, right, between the flexures, and this is going to produce another flexure, and this is going to be known as your pontine flexure. So again, mesencephalon, here we have our diencephalon and telencephalon, or your forebrain, mesencephalon, and then you have your hindbrain, and then here you're going to have your pontine flexure and your cervical flexure. And pretty much lastly, this is what you're seeing. Again, forebrain, mesencephalon, and rhombencephalon, or hindbrain. As you can see in your forebrain, you can have some of your cerebral hemispheres, your hypothalamus. Here we have our mesencephalon, and then in our rhombencephalon, right? Remember, our rhombencephalon is composed of our metencephalon and our mielencephalon. Just know that our metencephalon is going to be composed of your pons and your cerebellum, which are going to be here. So you have our metencephalon, pons and cerebellum, and you're going to have your mielencephalon, which is going to be composed of your medulla oblongata. And in the end, again, once more, you just kind of see how the brain undergoes all these kind of shape changes, but it's still the same thing. We have our forebrain, which is going to be composed of our ptelin and our dien. Here we have our telencephalon, and in our diencephalon, we have structures such as your pineal gland, your thalamus, your hypothalamus. We have our mesencephalon, which is our midbrain, and then we have our rhombocephalon, where you have our mielencephalon, and our metencephalon. Our metencephalon being our pons and cerebellum, and metencephalon, and your mielencephalon being your medulla oblongata. And this is it for development of the nervous system. Again, I did not cover every single detail due to time restrictions. I hope that you guys are able to understand the most important components, but definitely you guys are going to have to do some self-studying and make sure you know those other tiny important details.